Hello, welcome to today's Wednesday afternoon lecture at the National Institutes of Health. Thank you for coming. My name is Justin Taraska. I'm an investigator in the Laboratory of Molecular Biophysics in the National Heart, Lung, and Blood Institute. And it's my real pleasure to introduce Dr. James Rothman today as today's Wallace speaker. Dr. Rothman is the Wallace Professor of Biomedical Sciences and Chair of the Department of Cell Biology at Yale. Dr. Rothman is really one of the most innovative and influential cell biologists and biochemists working over the last few decades. During academic tenures at Stanford, Princeton, Sloan Kettering, Columbia, and now at Yale, his lab has done pioneering work in the field of membrane trafficking and protein sorting. This has included seminal discoveries related to how proteins insert into membranes, how vesicles traffic through the cell, and how vesicles actually fuse with the membrane, a process called exocytosis. Aside from these biological discoveries, Dr. Rothman's lab has invented many important methods, including the in vitro reconstitution of vesicle trafficking pathways, which has really allowed the complex steps of vesicle trafficking to be teased apart at their most fundamental level. He additionally developed novel pH-sensitive fluorescent proteins that have allowed individual synapses and even single vesicles to be watched in living cells in real time. In particular, Dr. Rothman has helped to establish the SNARE hypothesis of membrane fusion, which proposes that the correct pairing of alpha helical proteins on two opposed membranes actually directs and catalyzes their fusion. Along with these important discoveries, Dr. Rothman has mentored and trained many of the world's most prominent and successful biochemists and cell biologists. Dr. Rothman is a member of the National Academies, the Institute of Medicine, and the recipient of numerous awards, including the Lasker Award for Basic Science and also the Cavalli Prize for Neuroscience. Today, Dr. Rothman will discuss his recent work on SNAREs and the accessory proteins that direct their fusion in a talk titled Molecular Mechanisms of Synchronous Neurotransmitter Release. And I wanted to mention that after the seminar, there'll be a reception in the library, so please come, and it'll give you an opportunity to speak more informally with Dr. Rothman. So please join me in giving a warm welcome to Jim Rothman.
Thank you, Justin. Thanks for arranging the day, being a great host, and also sharing with me your very recent and exciting work, especially on the novel methods you've developed for measuring conformational changes in proximity in proteins with FRET. It's very exciting. I hope we get to collaborate as a result of that. So a pleasure to be here with you today. I'm going to, as the title suggests, talk about the process of synchronous neurotransmitter release. There's been a lot of progress over the last 5 years, I would say, especially in beginning to understand the mechanism by which this very important physiological process occurs in structural and biochemical terms. And so what I'd like to do today is offer some initial sort of historical background to the problem, then after that share with you our current view in the form of a model of how synchronous transmission neurotransmitter release may work, a structural biochemical model, and then after that show you some of the evidence that has accumulated for the model over the last 2 or 3 years especially. Well, what do I mean by synchronous neurotransmitter release? When it's actually the physiologists have various complex schemes to measure it and ways of defining it. To me, it's actually a very simple but really fundamental concept in neuroscience, which is when the action potential comes down the end of the nerve and you release a neurotransmitter across a synapse to the next nerve or perhaps a muscle cell, the neurotransmitter needs to be released at the right time. You don't want the neurotransmitter to be released asynchronously, meaning on its own accord, because in that case it's a false alarm. You also don't want the neurotransmitter to go not being released, because then you've missed an important signal. In fact, what you want is you want the neurotransmitter to be released precisely synchronously with the arrival of an action potential at a synaptic terminal, okay? The way that's achieved is through membrane-gated calcium channels, as probably everyone here knows, that are localized in the synaptic, pre-synaptic region that open up the gate for calcium entry, and calcium then acts as a second messenger to trigger the release of neurotransmitter that's stored in vesicles. The problem that we've sort of taken on and that I'd like to address here is, number one, how are these vesicles released? How do they fuse with the surrounding membrane, having stored the neurotransmitter within themselves? And how do they do it so very, very rapidly, so much more rapidly, orders of magnitude more rapidly than other membrane fusion processes that take place in the cell? And so those are the essential aspects that are built into synchronicity. Now, synchronicity is obviously important in a gross level, right? I mean, in your brain, if your neurotransmitters were being released helter-skelter, there would be no possibility of cognition, information processing, or anything of any remote interest, okay? You would have think about what the experience would be if your vesicles all fused, as they should, because the fusion proteins are constitutively active. They should fuse and release neurotransmitter all at once. So then you would have every neurotransmission event taking place all at once within a very short period of time. You would have no thoughts whatsoever, or perhaps every thought that you might ever have, and you would not be able to communicate it to everyone else. It might be an intensely transformational experience, but it would be one that would last about 10 milliseconds. So that clearly doesn't happen. But at a much more subtle level, the speed of synaptic transmission is very important for the complex circuits that we have. It typically takes a few milliseconds for a signal to be transported across a synapse, of which the release process, the initiation of it, takes typically less than a millisecond in central synapses. And that's actually very important, because from the time that a primary piece of potentially cognitive information, like a visual field or an auditory pattern or what have you, is sensed by our input-output devices, it has maybe 20 or 30 milliseconds before all those patterns coalesce at your highest centers, having been transmitted through perhaps 10 or 15 different synapses. I think we're all familiar with, some of us anyway are old enough to remember what movies used to be like, where the film would go by and you'd have 35 is the magic number frames per second, right? Why 35? Okay, because if it's faster than 35, it looks like a continuum to you. If it's slower than 35, you see separate pictures. The Hollywood folks were never spendthrifts. They were always looking for profit, and so they figured out first what psychophysicists learned later, which is that the granularity of human experience is about 25 or 30 milliseconds. And so you can get by with the least number of least use of photographic film at that speed, okay? So that 25 or 30 milliseconds is what you have that represents synchronicity, and the job is to get through 10 or 15 synapses and plow through them, from here and here up to here fast enough, okay? How does that work in molecular terms? And that's really the problem I'd like to address today. Now, the solution to this problem has come, I would say, over several decades, actually more like half a century, in which cell biology and neuroscience, or neurophysiology as it was then called, have dovetailed together, gone apart, come back together, and there have been many critical coming togethers of these two fields. The beginning of this field, in fact, reflected that. Going back to the classic work of Fatt and Katz, who found that postsynaptic potentials, this is the neuromuscular junction, in classic experiments from the early 1950s, if you have an electrical impulse stimulating the nerve, innervating the muscle, when you get synaptic transmission, the membrane potential of the muscle, of course, changes in response. But if you have no input, no action potential, nonetheless, on a much smaller scale, there are so-called miniature potentials, or minis, that appear at a fairly low frequency, but you can see that they're absolutely measurable, and they seem to be of a uniform size, measured as a postsynaptic potential. And that gave Katz the idea, the really transformational idea, that synaptic transmission, the release of neurotransmitters, occurs in a quantal fashion in which there are individual packets of neurotransmitter that are somehow prearranged, stored at a nerve ending as shown here. Now, at about the same time in the 1950s, my predecessor and the founder of the cell biology department at Yale, indeed the founder of, to a large degree, of the field of cell biology, George Palade, had observed these membrane-enclosed vesicles at nerve endings. He called these synaptic vesicles. And he also observed vesicles of many kinds in the cell and came up with the idea that vesicles are capable of membrane fusion. Well, not unaware of those concepts, Katz made the connection and suggested that these synaptic vesicles are in fact storing the neurotransmitter, a quanta of neurotransmitter, and made that connection in what became the vesicle hypothesis. The idea, of course, is that these vesicles fuse after stimulation, and it was also Katz who discovered that the entry of calcium into these neurons, synapses, is what provides or likely provides the immediate trigger. And in more modern work, and I'm taking you through the 1970s, in fact conducted here at NIH by Rees and Heuser, this vesicle hypothesis received very strong support where you could, they observed vesicles such as these that were containing the neurotransmitter, in fact were seen fusing with the presynaptic plasma membrane, releasing their neurotransmitter into the synaptic cleft to diffuse across the synapse. And so here then is one of the early coming togethers of cell biology and neurophysiology in the vesicle hypothesis. Now, this view that vesicles can store compounds for release from the cell, which, as Justin said, goes by the name of exocytosis, was independently reached and actually generalized by Palade and his colleagues. There are much larger vesicles that store hormones like insulin, or in this case, exocrine secretions in the pancreas, that are ready to be released and then are released following a stimulation. And what this introduces is a general concept, which is vesicles store products that need to be released rapidly, much more rapidly than they can be synthesized or localized to the site of release, and that this occurs physiologically. Now, in no place is this kinetic demand faster than in the brain, for the reasons that I mentioned. Probably second fastest is the release of insulin, which needs to occur in seconds to a couple of minutes. More leisurely is the process of membrane fusion as it occurs within the cytoplasm. And indeed, work in the 1970s and 1980s, as we began to discover the range of vesicles that traverse the cytoplasm, carrying cargo by budding and fusion from one compartment to another. We now recognize at least a dozen, and in some specialized cells, probably many more, types of vesicles, each of which selects for a cargo and then needs to deliver that cargo by membrane fusion. We came to realize that this process of membrane fusion is really very general, not just within a single cell cytoplasm. There are probably 10 or even 20 in complex eukaryotic cells, types of membrane fusion that occur, but also very general in biology, from plants to microbes, eukaryotic microbes, and of course to humans. And so again, by way of background, and this has already been introduced, what I personally find still quite astonishing is that this entire array of physiologically important membrane fusion processes, from the synapse to hormone release to the compartmental organization of the cytoplasm and its propagation in cell division, are all the work the handiwork of a single family of proteins. Now, this family of proteins, called SNARE proteins, comes in a number of varieties, but they have in common a single common mechanism, a physical-chemical mechanism for membrane fusion. There are two types of SNARE proteins that are physically complementary to each other. We call them V-SNAREs and T-SNAREs. They exist they're localized differently within the cell. A V-SNARE partners a T-SNARE and bridges the gap between two membranes, and as I'll describe in structural terms in a moment, not only initiates the process of membrane fusion, bilayer fusion, but actually fuses the bilayers. But there are many different types of V-SNAREs and T-SNAREs in a cell. They're shown here in different colors. So for example, the V-SNARE that is encapsulated in the vesicle that departs the endoplasmic reticulum can only mate with its cognate T-SNARE located at the entry face of the Golgi, and that is one of several molecular features, not the only molecular feature, but a critical molecular feature that dictates specific fusion of this vesicle here, not there, not anyplace else, and allows specific membrane traffic in the cell. Similarly, there's a different but structurally related homologous V-SNARE packaged into vesicles leaving the exit of the Golgi that finds its partner in the plasma membrane. And in fact, there's a different V-SNARE and a different T-SNARE depending on which surface of the cell, apical or basolateral. I could go on, but I won't. And so the specific pairing of these proteins between partnered membranes, including the release of vesicles at the cell surface as a special case, but a very important special case, occurs. Now, all of these processes, except fusion with the plasma membrane, are what cell biologists call constitutive processes. They occur all the time. They occur throughout the cell cycle. Yes, there may be regulation of one sort or another, there's no doubt, but they occur all the time. And this puts them into a fundamental distinction with certain classes of fusion with the plasma membrane, exocytosis, which only occur some of the time when a signal is provided, as in the release of neurotransmitter when calcium enters, secondary to the arrival of an action potential. The time course here is relatively leisurely. From the time a vesicle docks to the time it fuses typically takes 10 seconds to a minute, depending on the situation, but that's the time frame. Very different than the time frame in synaptic transmission, in which the neurotransmitter can be released as fast as 200 microseconds from the time calcium enters. And so that is the contradistinction here. And what confounds this further is that SNARE proteins, which I'll describe in increasingly greater detail in terms of how they fuse bilayers, are intrinsically powerful fusion proteins that are on rather than being off. So how can you have a neurotransmitter vesicle, synaptic vesicle, sitting right next to the presynaptic membrane with this potent fusion machinery that in and of itself is on, not off, and it somehow doesn't fuse? But then, at the right moment, it fuses very rapidly. So some answers begin to come from an understanding of the underlying principle of membrane fusion, which is encapsulated in the transition between these two structures. The SNARE proteins are alpha helical bundle proteins. They're relatively small. They have two parts, all of them, a part in the membrane, a transmembrane, and then they have a helix-forming so-called SNARE motif that is in the cytoplasm. The V-SNARE emanates primarily from the from the vesicle. That's why it's called the V-SNARE for vesicle, and it consists of a single helical protein, cytoplasmic domain. The T-SNARE, T stands for target membrane, which in the case of a vesicle fusing with the plasma membrane, the target membrane is the plasma membrane. The T-SNARE contributes 3 helices, and ultimately, when this unit assembles, it forms a 4-helix bundle. Now that 4-helix bundle is extremely stable, so that if you isolate this protein, this complex, from cells or from brain, or if you form it artificially with recombinant proteins, then we find, and others have found, of course, that you have to heat this protein to almost 100 degrees. You almost have to boil the water for it to denature. That's how stable it is. But there's a very interesting feature. These proteins want to fold to make a 4-helix bundle, but they can't do that when they're between two bilayers. And thus, it's from this that the principle of fusion follows. The V-SNARE is uniquely assembling from, let's say, the synaptic vesicle. The T-SNARE is uniquely assembling from the plasma membrane. As these two zipper up from their membrane distal end termini towards the membrane, they can assemble, but they can't completely assemble. I hope everybody can see that. Now, left to their own devices, for example, removed from a membrane entirely, they do fully assemble. In fact, even the transmembranes bind each other. And so they want to zipper up into this tight 4-helix bundle, but they can't. Why can't they? Because these two bilayers are separate. So only when these two bilayers become one bilayer can they zipper. So what we have here really is a thermodynamic linkage due to steric exclusion of two reactions. The first reaction is a protein folding reaction. The V-SNARE especially is a random coil, and the T-SNARE, though it has 3 subunits, is only loosely assembled before it encounters the V-SNARE. So in this state, the snares are partially unfolded or completely unfolded. In this state, after fusion, they are completely folded. In going from unfolded to completely folded, there's a lot of energy that's potentially released. If these proteins are simply mixed in a detergent solution or in water, they will give that energy off as heat. But placed between two bilayers, if they will provide enough energy to do work on the bilayer to cause the bilayers to fuse. Now, obviously, we don't understand by any means every detail about that transition, and there's a lot of important work that's going on, not the least from Josh Zimmerberg and others here. But there are a number of competing models, but what is now clear is that the assembly of these snares creates an inner force that pulls these two membranes together, and that force ultimately results in the opening of a fusion pore to release the neurotransmitter or some equivalent cargo. We know that this occurs with isolated SNARE proteins. The time course for this has been measured in a number of laboratories. It's less than 100 milliseconds. It's typically the average measurement is probably 30 to 50 milliseconds. So if you have isolated SNARE proteins with the V-SNARE and an artificial vesicle, the T-SNARE and a planar bilayer, that's the time course with which these snares will fuse. They are extremely competent. In fact, recent studies from Reinhard Jahn's lab, and we've confirmed this, show that a single SNARE complex that's assembling, we call it a SNARE pin, will be sufficient, at least at a certain rate. It is energetically sufficient to drive the fusion of a vesicle with a bilayer. So these are energetically competent. We have measured this inner-directed force directly using something called the surface force apparatus, where we can have the snares in opposite membranes, bring them together with sub-nanometer precision, pull them apart in a defined way, and measure the adhesive force. And indeed, there is an adhesive force, and it is a force and an energy perfectly consistent with the idea that a single SNARE pin is energetically capable of fusing a lipid bilayer. And finally, in case someone asks, at the end of this fusion process, there's an enzyme system involving a AAA ATPase called NSF that utilizes ATP hydrolysis to separate the snares, unfold them, and allow them to be recycled both energetically to the high energy state of an unfolded protein and also to initiate their recycling to the correct donor compartment. So this is our current understanding, in a cartoon level anyway, of how membrane fusion works. Now, to make this much more concrete, I'll draw upon a recent X-ray crystal structure of Reinhard Jahn and his colleagues in Göttingen that shows the 4-helix bundle. This is of the synaptic SNARE proteins. So by way of introduction, these are the proteins that do the job to release neurotransmitter at synapses. The V snare consists of a protein called VAMP, or synaptobrevin, and this is the one that originates in the synaptic vesicle. The T snare consists of two proteins, an integral membrane protein, which is called syntaxin, and a soluble protein called SNAP25, which contributes two of the helices. So the T snare here consists of three helices, two contributed by SNAP25 in green, one contributed by the membrane protein syntaxin from the plasma membrane initially, and the other, the vesicle protein VAMP, initially in the synaptic vesicle. And I want to distinguish three regions. There is a region called the bundle region, the helical bundle region, which you can see is called that for an obvious reason. Then SNAP25 terminates at that point, and VAMP and syntaxin continue to interact in a region called the linker region, which does not include contributions from SNAP25, called the linker, obviously, because it connects the four helix bundle to the membrane. And then you have the transmembrane domains of the V snare and of the T snare, VAMP and syntaxin. Now this, of course, is the post-fusion state that exists after the fusion. And if we want to understand the mechanism of fusion more, we need to understand more about the structure as it's assembling rather than after it's assembled. But this provides a very important guide. And importantly, it showed that VAMP and syntaxin continue to interact with a series of contacts into the bilayer even after fusion. So this zippering process evidently proceeds right the way through the fusion process. Now, if I didn't get anything across here, I'd like to sort of take a moment and summarize 25 years of my life in this one slide, which explains how I think about the problem. I hope nobody is offended for one or another reason. This is a lady's hairpin. And any of you who have had the experience of trying to separate the two ends of a lady's hairpin will know that it takes work to do that. So let's imagine that this is the snare pin that's assembling between two vesicles, except as we start out when it's already assembled, now we pull it apart. Now, having pulled it apart, we're going to do something a little bit unusual. We're going to insert each of the ends into a rubber ball representing two vesicles, or if you will, a synaptic vesicle and a plasma membrane, okay? And now what we're going to do is let them go. And as we let this go, what happens? The pin, because of its restoring force, inward directed force, that's the force that I'm referring to that occurs between snare pins. What will this pin do? It will force together these two rubber balls. Now, that really is the end of it if it's rubber balls. But if they're not rubber balls and they have a more liquid-like character, which in physical terms means they have a surface tension that can be overcome by the force exerted by the pin, the snare pin in reality, then the two balls will be blended into one, which allows the pin to reach its ground state and its minimum energy state. In my view, frankly, fusion is nothing more or anything less than this. There are a lot of debates about the transition state when those two balls are about to go together, and I tend to look at that as a statistical mechanical debate of interest. From a biological point of view, however, of greater interest is the thermodynamics that a precondition has been created that makes the fusion inevitable. Now, this example is not an idle one. In recent months, my colleagues and I have actually grabbed our hands on individual assembled snare complexes, zippered snare pins, and literally pried them apart with optical tweezers. So here, what Yonglei Zhang, a colleague in cell biology, and his lab and we have done is to attach a small bead by molecular modification to the assembled end of a snare complex, and we've artificially cross-linked the V and the T snare. These are the same synaptic snares you saw a moment ago, and we've artificially connected them at their membrane distal end termini. The membrane attachments would be here, but they're not. The transmembrane anchors are removed, and are replaced by a linker to one bead and a linker to the other on VAMP and on syntaxin. And now what we can do is exactly what I illustrated a moment ago and pull on them in a determined way. And what we observe is that as we do, they melt layer by layer. The first thing that melts is the linker layer. The second thing that melts is half of the 4-helix bundle. And they melt reversibly, so that as you pull, you get what a single channel physiologist would recognize as single channel behavior. That's because the linker has been destabilized, and at a certain force level it fluctuates between open, closed, and as it opens, the distance between the two beads increases, and that's what we're measuring here. Now, this will go on forever unless we exert more force, and when we get to the next force level, we are now able to melt this portion, which then fluctuates back and forth. Now, one of the beauties of this experiment, besides demonstrating actually the binary-like switch nature of these domains, is that it allows us to measure the kinetics with which this flipping back and forth occurs. And remarkably enough, for this synaptic SNARE complex, the rate of rezippering here occurs at what we estimate to be a diffusion-controlled limit. As fast as the V-SNARE can zipper in, as fast as it can lay down by displacing water, that's how fast it moves. This machine is not only energetically sufficient for fusion, but it is apparently designed to be as fast as physics allows, which is a wonderful fact when we consider how very fast synaptic vesicle release has to occur, again, the 200 microseconds. Now, I wanted you to note that there is a pause in the disassembly, so that we first bust this part, then we bust that part, and if we pull even harder, which is not shown here, is we pull this last part called the N-terminal domain, C-terminal domain, linker domain. N-terminal domain goes last. Of course, that's the guy that assembles first when the vesicle starts docking. That turns out to be very, very slow. It's very slow. It's measured, actually, its rate constant is measured on the order of an hour. And the reason it's so slow is that by being so slow, it allows other factors to kick in that can regulate the docking of the vesicle. And so what happens then is that you have factors upstream, and you'll see this, that regulate the docking of the vesicle to allow it to zipper through the N-terminal domain. And I'm going to give the story away now, because you'll see that in the special case of the synaptic vesicle, the vesicle is frozen at this stage, halfway zippered, punctuated in fact by what the crystallographers call an ionic layer. These are heptad repeats, and there's a certain hydrophobic pair in the heptad repeat that in every single SNARE complex is replaced by hydrophilic residues. This causes an intentional pause halfway through the zippering process, and in the synapse there are other proteins, and I will describe them to you, that grab the SNARE complex quite literally at this pause point and freeze it until the action potential arrives, until calcium arrives. And that is really nifty because it allows the SNARE complex to be activated from a vesicle whose frame is frozen just before membrane fusion can be completed at a diffusion-controlled limit. So that is sort of our picture of how this works. Oops. Now, to summarize then, there is, in this assembled SNARE complex, there are discrete domains, N-terminal domain, C-terminal domain, linker domain, and transmembrane domain. And they have distinct functions. The purpose of the N-terminal domain, which assembles first, is to dock the vesicle tightly to the plasma membrane. The next, the assembly of the C-terminal domain irrevocably commits the vesicle to fuse. The actual fusion occurs, as best we can judge, and I'm summarizing work principally from my lab and Reinhard Jahn's lab here, when the linker domain assembles. And then the transmembrane domain is believed to assemble based on the X-ray crystallography done by Reinhard and his colleagues. But since the fusion of the bilayers, as measured by the zippering is triggered by the linker domain zippering and the mixing of the lipid bilayers and the initial opening of a fusion pore, all of this would seem to be over, so there would seem to be no role necessarily for the zippering of the transmembrane domain. But actually nothing could be further from the truth, because, as we've recently published, the zippering of the transmembrane domain after fusion has occurred, after the fusion pore just opens, seems to be critical, based on in vitro experiments, for the opening of the fusion pore. That is to say, for the expansion of the fusion pore. And I won't really have time to go into this in detail, but if you're interested, this was just very recently published in Science. Lei Shi is the person who did the most work on this in the lab. And it involves kind of a novel assay in which we measure the fusion of SNARE-containing vesicles, artificial vesicles, with V-SNARE-containing vesicles with T-SNARE-containing nanodiscs. And the virtue of that is it opens a hole during the fusion process, so we can measure the release of content as a proxy for the opening of a fusion pore. And what we observe when we measure lipid mixing is that in no instance is there any big difference. You get lipid mixing with or without the various mutations that I'm about to describe. But when we go to Reinhardt's structure, when we mutate known contacts in his crystal structure between the VAMP and syntaxin transmembrane anchors, when we do that, we get very slow opening of the fusion pore, even though fusion has occurred with the normal speed. That's because the linker domains are intact, the bilayers have fused. Of course, you can't have topological fusion without having some opening of the fusion pore, so you get some release, but the rate of release is drastically increased. And if you mutate the non-contacting residues, we're showing only a couple here, but we've now done it quite exhaustively. Every molecular contact that's observed here is important for the opening of the fusion pore. So we're now turning to physiological systems to see if this is physiologically relevant, but it certainly suggests that each domain of the SNARE complex, including the transmembrane, has a critical role. So now I'd like to just encapsulate this in one more video. So here we have neurotransmitter stored in a synaptic vesicle. And the reason I'd like to show you this, even though it's a little repetitious, is going to orient you to what comes next, which is going to get a little more difficult. So here what we have is the T-SNARE in the plasma membrane. And in the initial state where the vesicle is docked, the N-terminal portion of the V-SNARE VAMP is in the helix. Its helical state, half zippered, through the helical bundle, so to speak, is halfway formed. But the remainder of the V-SNARE, including the C-terminal half that will form the bundle and the linker portion, has not yet zippered. And we believe, for reasons you'll see, that that is the point at which synaptic transmission is paused to allow synchronicity. Synchronicity, again, results because the vesicles accumulate at that stage and then therefore can be released synchronously. If they're not accumulated at a defined stage, they cannot be released synchronously. So it's really fundamental to the process. Now, the next thing that happens, this is in the absence of any regulation, just the general fusion process as we understand it, is that the C-terminal portion of the VAMP zippers, and this is what commits to fusion. Then the linker zippers, and as the linker zippers, this opens up the fusion pore initially, but only so far. And then finally, as the transmembrane zipper, this drives, we think it actually produces a radial force, though we don't really know this, it's just speculation, that literally forces the fusion pore open. Okay, so that's our view then of membrane fusion from the point of view of the proteins. Now I'd like to come back with all of that background to the very specific problem. Here you have a vesicle. It has, in vivo, a much higher concentration of V-SNAREs than in any of the in vitro systems. We might typically work with 5 or 10 VAMPs in an artificial vesicle of this size in vitro. But the V-SNARE here is present at 70 copies per vesicle. The T-SNARE, syntaxin, is present at perhaps one copy for every 1,000 phospholipid molecules in the artificial bilayers we use. Here, it's present in clusters that are almost pure with respect to syntaxin, according to the latest work. And so, remarkably enough, these vesicles stay where they are. They don't fuse, even though if you take the same proteins out of the synapse, if you put them into artificial bilayers at much lower concentrations, within 50 or 100 milliseconds, they've all fused. Now, I happen to be a believer in physics. I happen to believe that physics doesn't disappear when you put molecules into a cell. that actually the biophysics of molecules represents the ground truth of what a protein can do. And that ground truth doesn't change when it's placed, when proteins are placed in a cell. So I have to conclude from this that there's a clamp that blocks the exocytosis process and blocks it probably about a millisecond or less before the release process. What is that clamp? Well, over a 10-year period, we tried to find out what that clamp was, and in about 2005, we discovered what it was. It was a protein discovered by Tom Südhoff called Complexin. Now, Complexin is, as the name suggests, a complex protein. Sometimes names never fail you, even if they're terrible names. It was called a Complexin because it formed a complex with the SNARE complex, and nobody knew what else it did at the time. Tom knocked it out. It was needed for synaptic neurotransmitter release. In fact, we won't have time to go into this. It has both positive roles and negative roles. It is both an activator and an inhibitor, okay? But most importantly, I'd like to concentrate on its role as a clamp or an inhibitor, which we found when we added Complexin as a candidate clamp to fusion systems that contained the defined VAMP, syntaxin, and SNAP25 that otherwise fused, but if you added Complexin at a high enough concentration, they didn't fuse. Okay, so what did that mean? Not necessarily anything, but if we added the calcium sensor, which I'll come to momentarily, Synaptotagmin, back, and only then if we added calcium could we restore fusion in this artificial system. So that suggested that Complexin could be the clamp. Tom Südhoff and others since have done much more physiological experiments that show this beyond a doubt, and I'll show you a couple such experiments today. Now, Tom and Joseph Rizzo solved the X-ray crystal structure of Complexin in the early 2000s, and it's a helical protein. The helixes are probably the one thing you need to know about in this entire field, because in vesicle trafficking it's mostly about helices, including tether proteins. So Complexin binds to the outside of this 4-helix bundle in the membrane proximal half. Now, importantly, this is the structure of Complexin when it is bound to the fully assembled SNARE complex, which only occurs after fusion. It does not necessarily inform us, therefore, about the role that Complexin plays during the fusion process, where it acts as a clamp, okay? We added Complexin to this surface force apparatus experiment, which was a pivotal experiment here. Fred Poncet and I in his laboratory in Paris at the École Normale Supérieure. And what we found is that Complexin actually creates a new state in which the SNARE complexes are frozen in the half-zippered state, literally 50% zippered as best we could judge. So we knew that it did something important and it somehow could be the clamp. So the next question was, how does Complexin clamp? Now, as I warned you, I'm going to illustrate this to you by a model, and then I'll show you the X-ray crystal work the controls that stand behind it. Our present understanding, reached by Karin Reinisch, an X-ray crystallographer at Yale in the Department of Cell Biology in my laboratory, is as shown here. So this again is the half-assembled SNARE complex where the intervention by Complexin, we believe, begins. The Complexin helix actually has two parts, the central helix and the accessory helix. The central helix is endowed with the ability to bind both VAMP and syntaxin, and therefore binds to the assembled SNARE pin. And in fact, it binds to portions that are already present, but only present when the SNARE complex is half-zippered. So before this state, there's no binding by Complexin. When VN, as we call it, has half-zippered up to the ionic layer that I mentioned, then the binding site is created for the central helix. When that happens, the Complexin binds, and it binds in such a way as to leave the accessory helix pointing out. The central helix binding to the SNARE complex is very important because it means that you will always have a Complexin there. And by the way, remember I mentioned that this initial binding of VN to the T-snare is very, very slow. One of the factors that drastically accelerates it is the accessory helix. So this design ensures that the accessory helix, that the complexin is loaded at the time the vesicle firmly docks. So you are positioning your clamp in order to make sure, pre-positioning it to make sure it's there at the right time. The accessory helix, in fact, will be doing the clamping. If you take it off, and I'll show you some examples, you don't get clamping. If you mutate it, you don't get clamping. You need the accessory helix for clamping. You only need the central helix for the activation of neurotransmitter release. The next thing that happens is that the accessory helix does its job by reaching across and grabbing a second snare pin. So it clamps in trans. And in this way, one snare pin that's assembled clamps another, mutually inhibiting each other. Let's look at this from the top view, in which we are now looking at the proteins lying like a sandwich between the plasma membrane below and the synaptic vesicle above. So again, the first thing that happens is the complexin binds by its central helix, leaving the accessory helixes free to bind between snare complexes. Now it turns out the sequence of this accessory helix is homologous to the sequence of the V snare in the same region, which is very, very elegant. It's an honorary V snare, and look what it does. It goes right in here and it binds the T snare as if it were a V snare, but it's not. And by binding there, it prevents the V snare from further assembling. That unfolded portion of the V snare cannot convert to a coil, because to do so, it needs to displace the complexin. So there's a tug of war which results in clamping, so that if for any reason the complexin accessory helix is removed, as illustrated here, then the block to zippering is removed and fusion will proceed. But that doesn't occur because the accessory helix is clamping, this clamping occurring between two snare pins. Now, as you can imagine, this accessory helix, there's nothing to prevent it from forming another, and from recruiting another snare pin and another one to give rise to what we call a zigzag array, which is a highly cooperative structure, and that cooperativity gives rise, we believe, to synchronicity, because by coordinating a large number of snares in one structure that is all or none, has that all or none quality of a cooperative structure, it all will come apart at once. That's a clear prediction from the structure, and this will lead to the synchronous release of a vesicle. And so we think that ultimately is what synchronicity comes from. Now, this is how we regard clamping to occur. How does activation occur? So now what we've gotten into is a clamped vesicle. It needs to be released when calcium enters. Calcium, when calcium enters, the sensor for calcium is the protein synaptotagmin, discovered many years ago as a synaptic vesicle component by, again, Tom Sudhoff and also Reinhard Jahn. And in years of painstaking work, much of it controversial at various stages, but now widely understood to be correct, Tom went on to show that synaptotagmin is in fact the calcium sensor for synchronous neurotransmitter release. And probably the most persuasive experiment is a structure-activity experiment where he mutated the calcium binding sites in synaptotagmin to ratchet calcium binding up or down in terms of binding constant, put those into a mouse in place of the normal synaptotagmin gene, and was able to ratchet up or down sensitivity of calcium at synapses accordingly. So that doesn't tell us how calcium sensing occurs, but it certainly tells us that it does occur by means of this molecule. So how does synaptotagmin sense calcium? At this point, I'm moving from what I regard as fact to speculation. One thing that is a fact, though, is synaptotagmin is a tight binder of membranes. It has an aliphatic loop that actually inserts into the bilayer, and it inserts when calcium is bound at these calcium binding sites. In fact, the calcium binding site consists of a mixture of a compound of aspartic acid residues I don't think I've got a good picture of it here. Maybe I do on the next slide. Nope. There are aspartic acid residues here that bind to calcium ions and coordinate it with PIP2 that is on the membrane. So the actual calcium binding site is a sandwich of calcium between these two sites. Now, the cost of doing business for that calcium binding site is this aliphatic loop, which is inserted into the bilayer and must be inserted because that's the only way that the calcium binding site can be satisfied. So, synaptotagmin inserts. Mutations in synaptotagmin that abrogate this insertion likewise don't activate synaptic transmission. So one or another way, every model has to accommodate this as an important element of synaptic transmission. The other thing that synaptotagmin does in a large number of studies is perturb membranes in one or another way. The initial evidence here from Harvey McMahon was that in excess, when tagmin is added in excess to liposomes, it will actually tubulate the liposomes in the presence of calcium. That requires the insertion of this aliphatic region. Probably in less than megadoses it doesn't tubulate it, but physiologically it perturbs it. And every laboratory who studies this has found that synaptotagmin accelerates the rate of liposome fusion when you have SNAREs driving it in a calcium-dependent way. It does not fuse liposomes by itself, but it will accelerate probably a factor of 10 to 100, but very substantially the rate of lipid fusion, lipid membrane fusion. We don't understand exactly how that works. Probably it's by changing membrane tension according to Brunger and others, but it's another very important fact. So synaptotagmin is an accelerant. So it's both a calcium sensor and an accelerant. How does synaptotagmin fit into this story? Again, this is now I would say, the cutting edge of the field, but we kind of know how synaptotagmin binds to the SNARE complex. We know this largely from the work of Axel Brunger. I'll show you more details. And when it binds, it binds one per SNARE pin. And we imagine that it binds calcium from this array-bound synaptotagmin, and that when it binds calcium, it then inserts into the bilayer. And that we imagine further, this is our hypothesis, that I'll show you the side view, which is maybe a little more explanatory. So now you see the synaptotagmin binding. Now if calcium enters, or when it enters, and when it binds to a synaptotagmin, that will cause a rearrangement one way or another, which we believe exerts mechanical force on this array and removes a SNARE pin, thereby unclamping it. That's our model. The amount of evidence for it is relatively minimal, but it is, in fact, the simplest possibility. Now, where does all this come from? And I'll try to buzz through this much more quickly, not seeing the hour. Well, what Karen and I and our colleague, especially Danielle Kumel, the postdoctoral fellow who did the work here, and really quite a brilliant young guy, did is to solve the crystal structure of a half-zippered SNARE complex. You can see, for all the reasons I showed you, that when the vamp is half-zippered, that's kind of when we think all the action is occurring for clamping. And I explain why that is. Okay? Problem you have is you can't get a crystal structure of a disorganized state like a fusion intermediate or a half-assembled SNARE complex. So the next best thing you can do, which is what we did, is actually produce a stable half-zippered SNARE complex by using just the N-terminal portion of VAMP and leaving the rest out. Okay? So that produces a structural mimetic of a half-zippered SNARE complex. We were able to crystallize that in complex with Complexin, not synaptotagmin. No one's yet got a SNARE complex synaptotagmin crystal, to my knowledge. And we solved that structure. And we got something that was markedly different from what Sudhoff and Rizzo had found. Remind you, Sudhoff and Rizzo found that Complexin lies as an alpha helix in the groove between syntaxin and VAMP, contacting both of them. This is the central helix portion. And that an accessory helix they found had no contacts with the SNARE complex, but that this helix runs parallel to the SNARE complex. That's not at all what we found. We found a different conformation, which we called the open conformation, in contrast to the other conformation, post-fusion, representing the fully zippered SNARE complex that we called closed. Open goes off as 45 degrees. The Complexin helix is isomorphous. It's the same here and here, except for the way in which it lies on the SNARE complex. It's basically the same, but now goes off at 45 degrees. That posed a real problem for us because I was telling you all the genetics said that the accessory helix does the clamping. But what gets clamped? What gets clamped is the assembling of the membrane proximal, the membrane would be here, portion of the SNARE complex that assembles last. How can this interfere with the zippering of this when they're actually diverging away from each other? The answer comes from the crystal packing. When we look at a layer of the crystal, here now we see a SNARE pin. The membrane anchors would be at this end. The synaptic vesicle would be above, as in the cartoon. The plasma membrane would be in the plane below. Here's the half-zippered vamp. Okay? And now what you see is Complexin, central helix, launching an accessory helix. But that accessory helix in the open conformation actually binds in the same groove that this V-SNARE would if it continues to zipper, exactly as I cartooned it. So this is the basis for the zigzag array and the idea of intermolecular clamping as the basic principle for clamping of synaptic transmission to allow synchronicity. Now, any crystallographer will tell you this is very good, but how do you know that this isn't a crystallization artifact? Now, frankly, I'd never even heard of a crystallization artifact. It was a rude awakening. I thought as soon as you get a crystal structure, everybody congratulates you, publishes your paper. No, they send you back home. And that's where we biochemists actually start coming into the picture, because how do we know that this didn't occur as a result of the crystallization? Well, we know that it doesn't occur. How do we know? Well, because we can actually isolate complexes in solution, and we observe, to begin with, that this open conformation exists. So this was work done by Shyam Krishnakumar and Daniel Radoff in my lab. I won't go through the details given the time. We've used fret, intermolecular fret. We form complexes of Complexin and of the SNARE complex, and we put a fret acceptor on SNAP25 on the base of the SNARE complex, and then in two positions or more in the Complexin accessory helix, we place the cognate fluorescent probe, and this allows us to read the distance through fret. The fret signal will go up in the closed conformation. On the open conformation, it's less. And as you can see, we observe a very strong fret signal when we have the full-length vamp here, but when we have only the first 60 residues of vamp, the half-zippered state, now we get the open conformation. I hope that's clear, okay? And, you know, how do we know these conformations are what we say they are? We can take the fret numbers, the fret effect, we can crunch the numbers, and actually we predict within 10% systematic error the crystal coordinates of this position, this position, this position, and that position relative to that position in the two types of crystals, open and closed, in 3 different crystal forms. So we're very confident that this is the natural state in solution. Therefore, the crystallization did not cause the open state. The open state had to be accommodated in the crystal. Now, the other potential concern is how do we know that this trans interaction between the accessory helix and the T-SNARE at the V-SNARE binding site actually occurs independent of the crystal? Well, actually, we know that because we can measure it in solution now that we know to look for it. And again, this was published in 2011 in Nature Structural Molecular Biology. I need to update the slide. But if you mix Complexin and the SNARE complex with a half-zippered SNARE, okay, exactly what went into the crystal, but with its Complexin accessory helix site pre-bound and pre-blocked by a central helix. I'm sorry, I meant the central helix binding site pre-blocked, so that only this hypothetical trans site involving the terminal T-SNARE is available. We get nice stoichiometric binding, 1 mole per mole, 1.01, 15 micromolar binding constant, 10 kT binding energy, a number that's worth remembering, because that will turn out to be closely linked to the activation energy for membrane fusion. Now, very importantly, we've made mutations in the, I can see that, yeah. We've made mutations in the accessory helix that either increase or decrease the binding energy here. You can see them here. Oops. Here. So here's a mutation a point mutation in the accessory helix, right here, that eliminates binding of the in trans between the accessory helix and the T-snare. Here's a much more interesting mutation that actually increases binding energy between the accessory helix and the T-snare. So what would we predict? We would predict that this should not clamp very well. This should clamp fine because it's the normal gene. But this gene, tested physiologically, should clamp even better than normal and should reduce spontaneous fusion. So now I want to take you to the modern version of the fat in cats experiment in the hands of Richard Cho and Troy Littleton at MIT. So now we're measuring the frequency of Katz's miniatures at the neuromuscular junction. And this is what we see in a control animal, or what they see really. It occurs at a couple per second, more or less as Katz described. Now, if you knock out complexin, the rate goes up enormously, almost 2 orders of magnitude. Okay? That tells you that complexin acts, part of the genetic evidence, acts as a clamp in one or another way to prevent spontaneous release. A clamp, if you remove it, if you remove the clamp that prevents the spontaneous release, what happens to spontaneous release? It should go up. That's exactly what happens here. Complexin is the clamp. Now, if we start mutating residues that face the inside, the contacting regions for the T-snare, when we do that, then or if we disrupt the helix, now actually, and we put in the human complexin, we actually have no effect. Even though it's there, and it's there at the synapse, it doesn't clamp. If we put in the normal human gene, it clamps not quite as well, we're still trying to understand this, as the Drosophila gene in Drosophila. Maybe not a complete surprise because humans are not flies. If we put in the accessory helix mutant that has a 5 times higher binding energy, we get about 5 times lower spontaneous release. So to summarize then, this interaction in the crystal structure that we believe is the structural basis of clamping, that interaction must be controlling spontaneous release, because as we ratchet up and down that interaction, by the same logic as Tom Sudoff ratcheting up and down calcium binding, that ratchets up and down synaptic neurotransmitter release. That's why it's the calcium sensor. This must be the clamping interaction. If we ratchet it up and down, clamping gets ratcheted up and down in a physiological system. So there's no doubt that this trans interaction that gives rise to this zigzag array in the crystal structure is the structural basis of clamping, which must then occur in the half-zippered state. So this is a very important experiment, I think, for the entire case and for the entire model. Now, do we know that the zigzag array exists in this extended array way that it does in the crystal? Of course we don't know that. We're investigating that. It may or it may not, okay? It may be more truncated. Maybe it just involves 2 snare complexes. Maybe it involves more. I really don't know at this point. But that it involves, clamping involves this trans interaction seems very clear. But since there are 70 copies of VAMP and many copies of syntaxin around, it seems a reasonable likelihood that there are many copies that are engaged at a high concentration here as they are in the crystal. In fact, when we calculate the concentration of SNAREs in the crystal, and we estimate what it's like in the space here, it's actually lower in the crystal than it is in this space. So where you have 2 closely opposed membranes. So we think it's very likely, but we haven't proven that this array is of some significant extent, probably 10 or 15 copies of the SNARE pin, we're guessing. Now a very important point, and I will try to wrap this up now, is that this is the pre-fusion state in the half-zipper state. This is what everybody imagines a fusion pore must look like. It's got to be circular. If it has multiple SNAREs, they more or less have to be radially arranged, whether by intent or by or necessity. And the open state here of the complexin occurs, must go through a transition then between open and closed. There must be a transition to start that again, between the open and the closed state of complexin, because this is what we get when it's in the clamp state, and this is what we see, or what is seen, in the post-fusion state. So somewhere in there, there's a switch. Now, I think you can see that in order for example, this vamp to continue to zipper, that accessory helix has to get moved out of the way. Okay, that's sort of obvious. And, but when that occurs, okay, there's nothing then, when that happens, there's nothing to prevent this zippering. If you were to take somehow all of these open states and convert them to closed, I think you could see that the zigzag array could no longer form. The zigzag array can only form in the open conformation. In the closed conformation, the accessory helix is satisfied with its own SNARE complex and no longer can interact with another one. So this open to closed switch is clearly then what will be driving the activation process. Now, I won't have time to go into this. It's in our papers if you're interested. But it turns out that following residue 60 in VAMP, this is residue 1, there's residue 60, between residue 60 and 67, there are two very special turns of the helix in VAMP that contain 3 aspartic acid residues. We call them the switch aspartic acid residues. Those actually form an interaction with the accessory helix over here. That interaction cannot occur when those two turns are not yet folded. When those two turns are folded, they then create the binding site that is different between this structure and this structure, and they pull the complexin accessory helix down. So that what happens, in fact, we think, is that when any one of these accessory helices fluctuates out long enough, then this vamp, let's say this one here, will zipper. When it zippers 2 helices, that creates a binding site for complexin. Complexin is now switched from open to closed. When it does that, it retracts from this guy. When this retracts, then that vamp gets to zipper. Then it pulls down this guy, and like a house of cards, it all goes down. We've tested that by mutating the aspartic acid residues, and in fact, when we do that, you get in VAMP, and you get perfectly good fusion in and of itself, but you lose, at least in our in vitro systems, the ability to activate from calcium. We're now testing that in the physiological system, the neuromuscular junction. But in any case, that's our current view. So I just want to leave you then with our hypothesis of how activation may work, which is largely based on combining what I've shown you from our lab and very elegant work that Axel Brunger and his colleagues have done at Stanford, where they've used FRET extensively to try to measure the positioning of synaptotagmin on the SNARE complex. What's missing from the whole picture is a crystal structure of synaptotagmin bound to a SNARE complex in any state of assembly, and even a high-resolution EM picture. So if anybody's got ambitions in this field, that would be a very helpful thing to have. But based on his FRET and molecular dynamic modeling and a lot of biochemical data that's consistent with this, it appears that synaptotagmin binds to the SNAP25 side of the assembling SNARE pin. It binds 1 per SNARE pin. Stoichiometry, 1 synaptotagmin, 1 activator per SNARE pin, okay? And it binds to the SNAP25 face. There are 4 helices in the bundle, 2 on one side, 2 on the other. The 2 on one side are the 2 from SNAP25. They seem to bind the synaptotagmin. The other 2 is where the side is where the clamping occurs. That is where VAMP and syntaxin are and where the accessory helix, the central helix binds. So all of the action that I've just been showing you is occurring on the opposite side of the SNARE bundle from where synaptotagmin binds. Now, when synaptotagmin binds this way, very conveniently, its calcium binding sites are facing the membrane. This is shown with the synaptotagmin facing the vesicle membrane. In fact, we don't know whether it also or instead faces the plasma membrane, and we don't yet know which of these, if any, if either, is more significant functionally. So that all that needs to occur when calcium enters is for this synaptotagmin to reorient and bind the membrane. And we know that that occurs, and we know it's required for activation. And so the simplest model, since you know that from what I've shown you, that anything that removes a SNARE pin from the zigzag array, or its trans interaction more rigorously, will allow it to activate by removing the inhibitor, which was in the framework that it got removed from. Okay? Then the simplest model is synaptotagmin in one or another way carries this out. I won't have time to show you this data, not yet published, from Sham Krishna Kumar, but we formed SNARE complexes between two nanodiscs, which zipper up to a large degree but can't fuse because of the constraints of the nanodiscs. We've put Synaptotagmin on it, and then we've done stop flow, where we've measured on a scale of milliseconds, in fact, the insertion of this aliphatic loop of Synaptotagmin into the nanodiscs, triggered by calcium, and we've asked whether Synaptotagmin remains bound to the SNARE complex as it inserts. Because if Synaptotagmin is going to activate by physically doing work on the SNARE complex, removing it from the plane, its planar array structure, clearly it needs to hang on during this insertion process. And that is in fact the case. Here we're looking at fluorescence reporting the insertion of the aliphatic loop of both the A and the B domain. There are actually two domains, which occurs very rapidly after calcium mixing. And here we're looking at the distance between the A domain or the B domain and SNAP25, which is absolutely invariant. Okay? And by the way, also, for those of you who may be aficionados, the A and the B domain also don't move with respect to each other during this power stroke. So there is a power stroke in which Synaptotagmin inserts into the bilayer, clearly shown genetically to be needed for passage of the calcium signal to the SNAREs. It's physically bound to the SNAREs, and based on this in vitro work, remains bound to the SNAREs. And so the simplest and mainly, frankly, the only tenable model, because the Synaptotagmin is not even changing its conformation, A and B don't change their distance, is that a mechanical force is exerted on the SNAREs. And we can't yet prove today that that mechanical force is transduced into removing the accessory helix from the T-SNARE binding site that causes the inhibition. But obviously you can see that that's a very tenable hypothesis, and it's the one that we're focused on. And that hypothesis is tenable both energetically and kinetically. This will be my last slide. Energetically, do you remember the number 10 kT I asked you to remember? The activation energy to remove a snare pin will require breaking two interactions. It's interacting with one snare pin and in this array with another. Okay? And this interaction could be broken either by breaking the accessory helix interaction or the central helix interaction. Central helix binds with about a nanomolar. This guy binds with about 10 micromolar, so you can guess which one is going to break first. So 10 kT for this interaction, that will break this one, or 10 kT will break this one. If you pull out, if you have a fluctuation, if you have 10 kT only, you'll get fluctuations here on the order of about 2 microseconds, according to single molecule types of calculations down here, according to the somebody or other relationship, which my physicist colleagues tell me about, and which I might once have understood. But anyway, sorry to be blasé about that, but the physicists all agree that this fluctuation occurs in 2 to 20 microseconds, which isn't long enough to be persistent. So that's what allows determined activation by removing 2 of these points. When you remove 2, when you look at the lattice, you will see that the whole thing falls apart like dominoes, but if you remove only 1, it can re-anneal. And so it takes about 20 kT, probably, to cause activation. So the simplest hypothesis is that this snare pin, for example, could be any of them, is being ridden like a horse by a single synaptotagmin. And when that lucky synaptotagmin gets loaded with its complement of calcium, it then moves out of the plane. And in fact, this zigzag array, I haven't emphasized that, is a rigidly planar structure. The contacts are broken. If you pull it out, it opens up. And according to this hypothesis, inserts. So the total activation energy should be 20 kT. Fortunately, Reinhardt and his colleagues have measured the activation energy, the actual energy released when synaptotagmin binds calcium plus PIP at the relevant concentration of calcium. It's 45 kT. Does that mean we know that 45 kT is fully available to be channeled to this? No. It does not know that, but we don't know that, but at least it's a tenable hypothesis, and kinetically it's tenable as well. So finally, sorry for the breathless finish, I'd like to just thank the colleagues who are most critically important for this work, the X-ray crystallography, Danielle Kumel and Karen Reinisch, a professor in the Yale Cell Biology Department, the electrophysiology that was so important in establishing the physiological relevance of the structure, a collaboration with Troy Littleton in the hands of Richard Cho at MIT, The initial work that got us going in the first instance, which is the discovery that Complexin clamps in a half-zippered state that gave us the clue to how to design the molecule for the crystal structure. Frédéric Poncet from the École Normale Supérieure in Paris is a physicist and has been very important in all of our collaborations. And more recently, the optical tweezer work with another professor at Yale, Yonglei Zhang, and the folks from my lab who I've mentioned, I believe, as have gone through the work in their particular contributions. So thank you all very much. I've been told to stand here at the X several times, so I guess if people have the patience to have a Q&A, I'm happy to do that.
So in the interest of time, if you'd like to go, and we'll take questions after the hall is sort of cleared out of the people who need to go right away.
Okay.
So.
Yeah, no, I don't mind. I'm happy. It's either here or go over to the reception. It'll be harder to do that. So.
Take questions?
Yes?
Thank you. Very, very
Show me where.
fascinating.
Oh, there you are. Okay.
Very fascinating, intellectually provoking lecture. It reminds me of what you mentioned as far as the time course of the signal transduction. That, as you also mentioned, it happens with hormonal, and also it happens in the immunological, except for slower pace. My question and maybe a suggestion is, you mentioned the N-terminal is actually starting the whole game for the structure of and the signal to the N-terminal. Is there any information known about the N-terminal composition? For example, is there a sulfhydryl group that it's...
I'm not sure. So what's the question then?
The composition of the N-terminal and the C-terminal changes, and the charge, charges...
Maybe that's something we should discuss in the privately, but the composition of the amino acids isn't very likely to change, if that's what you mean. Is there another question?
Okay.
Yes.
It seems like one of the critical things in the measurement of these synchronous phenomena and time-dependent phenomena are the tools that you use. And things like X-ray crystallography are really a snapshot of some phenomenon that happens over a period of time, whether it's femtoseconds or nanoseconds. If you had a wish list of a new tool that you could use or design, or maybe there are some in existence, what would that tool look like? Optical tweezers or some other tool
Well, me, I'd like to miniaturize myself in so many ways. And if I were only on the atomic scale, I would give you all the answers, but you'd have to really listen up. I'm not sure.
I am curious, besides PIP, is anything known about the local lipid environment and how that's perturbed at the synaptic junctions and how that might assist this whole vesicle fusion process?
Yeah, that's a really good question. Well, a PIP, aside from PIP, is a big aside. And that PIP is absolutely required physiologically. It's required in the in vitro systems for the maximum effect. It's very, very important. Okay. Other than that, we're sort of unfortunately somewhat left with the imagination. The SNAREs are largely uncaring about the lipids. I mean, the speed will go up or down somewhat, a factor of 2 or whatever, but not determining. But there probably is something very important in the clustering. The T-SNAREs seem to be in many different cells in so-called lipid rafts or domains that are cholesterol dependent. We don't know enough about them. So, I don't want to say that I actually do think they're actually quite important. Maybe the active zone itself is a lipid raft. There's some suggestions along those lines. So I think there is a vital importance, but the neither the in vitro systems nor the sort of crude genetic tools that are available, I don't mean to say genetics is crude, but relative to this type of physiology, that knocking something out, adding a lot of it, I mean, it's all relatively crude. So we just don't know. I'd like to know.
Do you have any insights into what happens in asynchronous release, which may be important for insulin
Yeah, something
secretion,
that
for
doesn't
example?
require synaptotagmin.
Does not require synaptotagmin.
Yeah, so I mean, I don't have any personal knowledge of that. My impression from reading the literature is that synaptotagmin is not the calcium sensor for that. My impression is that there's some sentiment that there's a related protein called DOC2 that may be the calcium sensor, but my other impression is that it's probably still somewhat controversial. But there is a calcium sensor, that's clear.
And what is Complexin doing in that situation?
I don't know. I would predict probably nothing, okay? I would guess it's not involved. And in fact, you can get calcium dependent. In fact, the nicest experiment here, I should say the nicest, there's a lot of nice experiments, but to my mind, the nicest experiment is an in vitro experiment with pure proteins that was published by Thomas Solner last year, because you have absolute control of everything. And what he shows is that if you leave Complexin out, okay, but you still have PIP2, synaptotagmin, and the SNAREs, you will get vesicle docking, you will get release that's calcium dependent, but the amount of fusion in the absence of calcium goes up very dramatically. The amount of vesicle fusion that you get when you add calcium is, it brings you up to 100%, but there's less far to go because you already fused 20% of it, so there's less release, if you want to think about it that way. And instead of it getting done in less than a second, it dribbles out over 10 or 20 seconds. Add back Complexin, no fusion in the absence of calcium, totally clamped, okay? 100% release, and all within the first second. So asynchronous goes to synchronous. So if it's asynchronous, depending on how asynchronous it is, you can probably get that with the synaptotagmin alone. And I think you could see from the way synaptotagmin is positioned that it itself is likely to be an impediment to membrane fusion by sitting between the two membranes. Okay? So that's probably, it slows the terminal zippering by being in the way in some way or other. We haven't yet sorted that out, but I think there's probably going to be a fairly simple mechanism for that.
Well, great. Let's adjourn to the library, and thank you all for
Thank
coming.
you.
So thank you, Doctor.